Dr. Johnson here, and as promised, today we're going to explore the science behind antibody testing by running an actual test. I've got some patient samples here on hand, and we want to determine if they've been infected by the coronavirus. We can do that by looking for specific antibodies in their blood. Remember from our last video, antibodies are those Y-shaped proteins that our bodies make in response to infections caused by pathogens like the coronavirus. So how does it work? In general, an antibody test takes advantage of how antibodies bind to antigens during our immune response and replicates it inside a plastic testing container. But we can't see if antibodies are there, so we use a special enzyme-linked antibody that will speed up a chemical reaction and result in a color change and tell us if those antibodies are present. Before we get started, I've got a quick disclaimer. None of the reagents that we're using today contain live virus or real patient blood samples. Everything we're using is being strictly used for educational purposes only, so no need to worry. So here's our testing container made of polystyrene plastic. The first 3 wells are labeled with a positive. These are our positive controls. The next 3 are labeled with a negative. These are our negative controls. Then we have 3 labeled with the number 1 and 3 labeled with the number 2. These represent samples from our 2 patients. We're running each sample 3 times so we can be confident that the final result is valid and also account for any human error. Let's do the test. For step 1, we're gonna add antigen to all 12 wells of our testing container. Remember, we have to provide the antigen in order for the antibody to bind to. And so we're gonna give it 5 minutes to incubate. And basically that's allowing enough time for the antigen to stick or attach to the surface of the polystyrene well. And it does this through hydrophobic interactions. That incubation time is up, it's time to use our wash buffer. Remember, that's our science soap or detergent that helps remove any molecules that haven't stuck to our polystyrene well. So these floaters right here, when we add that wash buffer in and pour it out, they are gonna go away, leaving just the ones that have bound or attached to the well. The wash buffer also is going to coat the inside surface of each well, preventing any other molecules from binding to it. This is going to help prevent false positive or false negative test results. Now it's time to add our samples. In our positive wells, we're purposefully adding in those antibodies because we want a positive test result. In our negative wells, we're purposefully leaving these antibodies out, so there won't be any antibodies present because we want a negative test result. Controls help us do 2 things. 1, they let us know that the test is working properly because we know what the expected result should be for those test samples. 2, it also gives us a visual of what a positive or negative test result should look like, because when we add in our patient sample, we don't know the outcome. We're only going to be able to determine that once we get to the very end. Again, we're going to let our samples incubate, so those antibodies, if they're present, will make their way down to the antigen and bind just like they do in our body. Now it's time to wash again. Remember, we're doing this to get rid of any of the unbound molecules. So in this case, we want to get rid of these antibodies that haven't found an antigen to bind to. So when we add that wash buffer in, these guys are going to go bye-bye. Now we're adding in that enzyme-linked antibody. That's that lab-generated antibody that has an enzyme on top. That enzyme, the little Pac-Man shape, is gonna help speed up a chemical reaction that's gonna let us know whether or not these antibodies are present by causing a color change. But not quite yet, we've got 2 more steps to go. Now it's the final wash step. Remember, we're using this to help get rid of any unbound molecules. So we've got a few of those enzyme-linked antibodies that aren't attached to anything. So when we add that wash buffer in, it's gonna come in and get rid of all of those unbound antibodies. For our final step, we're gonna add in our enzyme substrate. That's our starting material or our reactants for our chemical reaction. They're gonna float down and they are going to bind or attach to the enzyme. That enzyme is gonna help speed up that chemical reaction, which is gonna turn those reactants into products. And in this case, the product is gonna cause a blue coloration. So in these next few images, I want you to start to watch the wells and see, do you see any blue? So what can we conclude from our results? Well, if we take a look at our container, we can see that our positive wells are nice and bright and blue. meaning yes, antibodies are present. And that's good because that's our positive control, so it should have turned blue. Our negative control stayed clear, meaning there are no antibodies present, which again is good because that's our negative control and that's what we expected. Now, if we look at our patient samples, the wells labeled 1 and 2, we're seeing two different stories. So for patient number 1, their results matched our positive control, which means yes, antibodies are present. Versus patient number 2, their results match our negative control, meaning no, there are no antibodies present. So we can conclude that patient number 1 has been exposed to the pathogen because the only way their body is going to produce those antibodies is if they've been infected. Patient number 2 has not been exposed to the pathogen because they don't have any antibodies present. So what can antibody tests tell us? Well, since not everyone who's infected shows symptoms, testing for antibodies may help healthcare professionals know the true infection rate of the coronavirus. Doctors can also use this information to study what happens to patients who get infected a second time. Remember that secondary immune response? Which can help us better understand immunity to COVID-19. And lastly, it can inform us about herd immunity. This is when the majority of the population has antibodies to a particular pathogen, like the coronavirus, and it helps protect those who aren't infected. However, scientists are still determining if this will be the case with this particular coronavirus. Thanks so much for tuning in. Who else loves science? Antibody? Antibody?